So Streptococci is a gram-positive cocci organism. Gram-positive organism which looks blue in color in under the microscope with gram staining, blue or violet. So strap means linear in shape, whereas cocci means round or oval organism, oval shaped organism, oval shaped. So Streptococci under microscope it looks like round organisms which are organized in chain-like fashions. Sometimes we can see two cocci together, or sometimes we can see chain of organisms which are blue or violet in color, as seen in gram staining. So there are 30 species of Streptococci we can see. So in this 30 species, very few can cause infections. So how we can classify? C carbohydrate, which is present on the cell wall of these organisms. And the second one is hemolysis. So first we'll see Landsfield classification, how we can classify this Landsfield classification. Depending upon the C carbohydrate, these Streptococci, the C carbohydrate structure, there are A to S varieties. A to S varieties, depending on Landsfield antigen. Landsfield antigen. So we can analyze here. Landsfield antigen, A to S varieties. But these A to S varieties, not all these A to S varieties can't cause infection in humans. Only few varieties can cause infection. So what are those few varieties? That is Streptococci A group. Yes, A group is nothing but the C carbohydrate which has a structure of A. So the C carbohydrate which has a structure of A, we can call them as Streptococci A group organisms. That is one. Second one is Streptococci B group. So that means this B group of organism will have a C carbohydrate structure as B. And the third one is Streptococci D. So this D group has the structure of C carbohydrate as D structure. So only these A, B and D normally causes infection in humans. They are pathogenic to humans. Pathogenic to humans. So there are non-typable streptococci that also can cause infection. That means they won't have a C carbohydrate. Those non-typable streptococci are one is virulent streptococci. So that means these organisms, they won't have the C carbohydrates on their cell wall. And the other one is Streptococci. So some people will call it as cocci, some people will call it as cocci. So anything is fine. Streptococci pneumoniae. So altogether, the streptococci which can cause infection in humans are these 5 varieties: group A streptococci, group B streptococci, group D, Viridin streptococci, they won't have a C carbohydrate, and Streptococci pneumoniae, which won't have a C carbohydrate either. So depending upon landfill antigen, we have 5 varieties which can cause infection. Whereas coming to the hemolysis. So depending upon hemolysis test. So first we know what is hemolysis test. Hemolysis test is nothing but it is done on blood agar culture medium. So whenever we have a blood agar culture medium, if suppose take example, if this is a petri dish which consists of blood agar culture medium. So on this petri dish, if the organisms which are grown on this petri dish, they completely destruct the red blood cells entirely, completely together, we can call that hemolysis, destruction of complete red blood cells, clear-cut destruction, which most of the time looks clear in nature. The colony looks very clear in nature around them, something like this. So we can call these colonies as beta hemolytic colonies. So any organisms which are grown on this blood agar culture medium, by destructing the complete red blood cells, clear-cut destruction of red blood cells, we can call those colonies as the organisms which are growing by completely damaging the red blood cells as beta hemolytic organisms or colonies. Alpha hemolytic. There is one more pattern called hemolytic test pattern called alpha hemolysis. These alpha hemolytic colonies, they always have a partial destruction of red blood cells. Partial destruction. Something like this. The organisms will grow on this blood agar culture medium, but they won't completely damage the red blood cells on the culture medium. They partially damages the red blood cells, leaving green color hue around the colonies. The colonies looks light green in color because of the remnants of hematin always gives light green color around the colonies. We can call that partial hemolytic pattern as alpha hemolysis. Alpha hemolytic colonies. And there is one more pattern, that is gamma hemolysis. This is a petri dish, and these organisms, they grow on this culture medium. As white color colonies on this culture medium without destructing any red blood cells. That means something like this. Which is gamma hemolytic colonies. So, coming to our Streptococci classification depending upon hemolysis, we have as landfill classification, we have A, B, D, Viridin Streptococci and Strep pneumonia. These 5 groups are causing infection in humans. Whereas Strep A, Strep B are beta hemolytic. Whereas alpha hemolytic ones are Viridin, Strep pneumonia, Streptococci pneumonia. So these are alpha hemolytic. Whereas gamma hemolytic ones are, the last remaining one is Strep D. Strep D, Streptococci. So depending upon hemolysis, we have beta hemolytic organisms are group A Streptococci, group B. So these two organisms are beta hemolytic. That means they destruct entire red blood cells on the blood agar by clear-cut destruction. Whereas Viridin and Strep pneumonia, they cause partial destruction of red blood cells. They can't destruct completely. So you can see a green color hue around the colony. Whereas gamma hemolytic colonies, they never destruct the red blood cells. So Strep D comes under gamma hemolytic. So depending upon this classification, we have 5 groups which can cause heterogenic organisms. We can classify with landsfield antigen, and we can also classify with hemolysis. So that's about the Streptococci classification. Thank you.